Okay, so I'm Katerina Pony, and this is my presentation on counterintuitive schizophrenia alleles, mutation intolerant yet faced by background selection. So, schizophrenia is a debilitating psychiatric condition that is both highly heritable and polygenic, which means its genetic architecture involves a continuum of variants with small effects. The relative contributions of these alleles of various frequencies is yet to be resolved. Recent studies show alleles explain actually up to a third to a half of the genetic variants responsible. This means that variants of the genes that boost schizophrenia, though by small amounts, are actually fairly common. And as a result, there's been a long-standing debate as to how the common risk alleles are able to persist in the population, particularly given the mean reduction in life expectancy of 15 to 30 years and the decreased fecundity associated with schizophrenia. And that's as a result of the poor social and functional outcomes. The truth is, the genetic origin of schizophrenia is unknown, and the many theories, which include things like allele hitchhiking and balancing selection, can now be better addressed given the advances of technology and also importantly, the social attitudes to mental health. Unfortunately, there has been no substantial improvements made other than antipsychotic medication in the mid-20th century, and that's definitely attributed to the lack of knowledge of pathophysiology. So Pardiñas et al. Set out to find more information on specifically the schizophrenia causing alleles to better understand the mechanisms by which common risk variants are able to persist in the population, and to importantly contribute valuable knowledge to the pathophysiological fields of science. They found that mutation-intolerant genes are linked to risks in developing schizophrenia, that mutation-intolerant genes also account for a large portion of SNP-based heritability, and I'll go into detail about these, but SNP stands for single nucleotide polymorphisms, or also known as variants. They also found that background selection is a potential cause for the high frequency of causal alleles in the population, and background selection is referring to a selective pressures that aren't tightly linked to the gene of interest, or simply just the loss of genetic diversity at a non-deleterious locus. So they also highlight 6 particular gene sets that are independently associated with schizophrenia, and they also identify potential causal variants. And these findings are interesting, but for one particular reason is that mutation intolerance is known to be due to high selection pressure. Therefore, findings that schizophrenia risk variants that persist at common allele frequencies are enriched in mutation-tolerant genes, and also the evolutionary pressure appears to be background selection, all appears counterintuitive. So how did they do it? So firstly, they did this by running a GWAS, and that stands for Genome-Wide Association Study. Genome projects primarily investigate the relationship between phenotype and genotype, and a GWAS is no different. It's a study with usually quite a large data set, and it runs gene identification estimations of genetic correlations between traits. So what this means is that firstly, you have to sample the population for cases and controls. Here they obtained 24,542 controls and 11,260 cases. And they also obtained data from the PGC, and that's the Psychiatric Genomics Consortium. And that's just sort of for a meta-analysis style study, and that total amounted to over 64,000 controls and over 40,000 cases. And that's all in order to increase statistical power to detect associations in the meta-analysis. Largely, sample sizes are important in explaining the heritability of traits. That's why it's quite important for a large sample size. So secondly, as a GWAS study goes, you need to genotype the sample, and in this case, they used an SNP array, SNP being single nucleotide polymorphisms, rather than whole genome sequencing, largely because an SNP array is cheaper and more commonly done at the moment for GWASs. Thirdly, as you would in a genome-wide association study, they run a statistical association to test for significant associations of loci within the chromosome. And I'm going to go into detail about the results. So sorry, I think my computer had some notifications. Their statistical association, which you can see here in this in their Manhattan plot, identified 145 genome-wide significant loci. The red line represents the significance threshold. So loci surpassing it into the space above were considered significant, and these are also labeled in green. So across these 145 genome-wide significant loci, they observed that mutation-intolerant genes were shown to obtain large numbers of the SNPs, the single nucleotide polymorphisms. And what's interesting is that mutation-intolerant genes are assumed to hold critical biological functions and are actually present in rare variants in psychiatric disorders. So to measure the SNP contributions or impacts on mutation-intolerant genes on schizophrenia, they measured the schizophrenia SNP-based heritability. So they applied a heritability analysis of the gene sets. What SNP-based heritability is a measure of the proportion of SNP's variation that is caused by genetic variation. And in order to measure this contribution within mutation-intolerant genes, they applied something called a linkage disequilibrium score regression, which I'll refer to as LDSR. An LDSR measured the contributions of polygenic effects and confounding effects, such as environmental or selective factors. It's a regression analysis of the relationship between non-random associations of alleles at different loci and SNPs. So their results showed that the genic SNPs accounted for 64% of SNP-based heritability, and that mutation-intolerant genes accounted for 30% of SNP-based heritability, and that makes up almost half of all the genic SNPs. So using LDSR again, they observed strong evidence for schizophrenia SNP's heritability under strong background selection, and I'll go into detail about that. So they did this by partitioning the LDSR test in order to observe the relationship between schizophrenia-associated alleles and SNP-based signatures of natural selection, and those are background selection, positive selection, and Neanderthal introgression. They observed that SNPs were significantly enriched under background selection, which you can see I've circled in the big red circle for that in the table. And it was more significantly enriched than any other natural selection metric. And this was also consistent across all the thresholds examined. And they examined quite extreme values of thresholding, 2%, 1%, and 0.5%, and that's just for statistical power. Interestingly, there was also observed a depletion of SNP-based heritability in SNPs subject to positive selection. So I've circled that in, it's a smaller circle just below. And what's also interesting is that the metric above, which is the positive selection calculated with SLR statistic, is a stat that controls for the effect of background selection. So even controlling for the effect of background selection, it's still got a lower enrichment score. So to then illustrate this mechanism of selection more clearly, they binned the background selection intensities into 4 categories of increasing score and classified SNPs into these bins. So I'll go into detail with that as well. They did this according to whether they were genic or non-genic and whether they were mutation intolerant, other genes or other gene sets or CNS related, so central nervous system. In this diagram that illustrates the heritability of genomic partitions, the radius of each segment indicates the degree of enrichment, and the arc, or just the angle of each segment, indicates the percentage of total SNP-based heritability explained. Here you can visually identify that the heritability enrichment of genic SNPs is quite substantial, but also that the CNS-related and mutation-intolerant genes make up a large portion of both enrichment and SNP-based heritability, SNP being single nucleotide polymorphisms. So Pardiñas et al. Starts to suggest that background selection and mutation intolerance are independent contributors to the heritability of schizophrenia alleles. So they carry on, and it's required to do a functional characterization of genetic variants to move from simply statistical associations to actually identifying potential causal variants or actual genes. So they decide to focus the analysis on CNS-related genes on the central nervous system. So they compiled 134 gene sets that were related to different aspects of CNS function and development. They found that 54 were nominally significant and wanted to continue narrowing that analysis down. So they applied something called a stepwise conditional analysis, and that's where you just adjust sequentially for the more strongly associated gene sets. And they found that 6 gene sets were independently associated with schizophrenia. So the most strongly associated was one particular gene set, the Fragile X mental retardation protein known as FMRP. It's been associated with autism before and has been recorded as nominally significant for schizophrenia before, but that's been quite inconsistent. So this provides some stronger evidence thus far for the presence or maybe impact of the FMRP gene set. So of the rest of the 6 gene sets, the 3 derived from the mouse genome informatics database and relate to the behavioral and neurophysiological learning and abnormal nervous system electrophysiology and abnormal long-term potential. And then 2 gene sets were independently associated with gene sets of voltage-gated calcium channel complexes and the 5-HT2C receptor complex. So to identify SNPs and genes that might be causally linked to genome-wide significant associations, they used fine mapping. And fine maps are just an additional process in GWAS where regions are analyzed to identify particular genetic variants that are likely to have causal influence. And I've got an example of what that might look like on the image at the bottom right, but that isn't from this study. That's just an example. Unfortunately, they didn't have one for this study, an image of it anyway. So they identified 6,105 credible SNPs across 144 genome-wide significant loci. They did this using chromosomal conformation analysis performed in tissue from the developing brain, and they were able to narrow down to a highly credible set of 25 SNPs, as those that were just normally likely to explain the associations than all other SNPs combined. So of those 25, 14 mapped to genes, and finally, one of the implicated alleles was a non-synonymous variant in the manganese and zinc transporter gene SLC39A8. Variants of this gene, the transporter gene, lead to SLC39A8 deficiency and have been associated with other neurodevelopmental disorders. So this finding is potentially a therapeutic target for schizophrenia. But I think there are some caveats to consider about this study before I also talk about the impact on modern science. So on more general terms, within the scientific community, there are some concerns that most association signals reflect variants in genes with no actual direct biological relevance to the disease. And that's because critics contend that SNPs identified in GWAS explain only a small fraction of heritability of complex traits and do not necessarily pinpoint to causal variants in genes. And they also simply yield too many loci, and that just becomes uninformative. SNPs also have a bias towards variants discovered in well-studied or sequenced populations and are mainly restricted to common and low-frequency variants. But it is also important to note that they're getting better. As technology increases or gets more efficient, rare variants are slowly getting more accurately noticed. So most genome-wide association studies use SNP assays as the method of genotyping. But for identifying causal variants, it's actually much easier and efficient to use WGS, whole genome sequencing. However, currently in time, WGS is extremely cost inefficient, and as the cost of WGS continues to decline, genome-wide association studies will start using WGS more, and in combination of larger sample sizes and improved computing, a shift to whole genome sequencing will let genome-wide association studies account for population stratification, and then you'll be able to identify ultra-rare mutations and more. So also, one of the 6 gene sets identified associated with the 5HT T2C reporter complex should also be accepted with caution. And I'm going to quickly explain why I think that is. So this gene site has already been a previous avenue of schizophrenia research and is now considered just ever so slightly outdated. You can see on the table on the left, I've labeled it 1. I've circled the number of genes in it. It's also quite small, so it's got 16 compared to a few hundred or a few thousand. And it's also mentioned in the paper that a number of its genes encoding proteins structurally also relate to other receptor complexes. So that's why I think if you have a look at what I've labeled as number 2, there's an extremely small proportion of genetic heritability explained by this gene set. And then also, if you look at number 3, I also think that's why there's a huge amount of variation, because you can see the error bars are huge. So I think it should just be accepted with a bit of caution. Okay, so impact on modern science. Nonetheless, despite all the caveats, I think that this is an important study. Studies like this in general are important for diseases which can be for diseases such as this, which can be characterized by such negative and impactful symptoms. For modern science, this study provides, importantly, more genome-wide association study data. It's only in the last decade that GWAS have been popularized and revolutionized the fields of complex disease genetics, particularly in a world that lacks knowledge of pathophysiology. It provides some explanations on the genetic architecture of schizophrenia, and in short, that being some novel gene sets to focus on, some more statistical evidence to help confirm associations with mutation-intolerant genes, some mechanical explanation with evidence for background selection, and finally, novel causal variants. On simply a scientific standpoint, these findings are highly interesting to better understand genetic conditions such as schizophrenia and their origin. However, in a clinical setting, insights into the architecture of schizophrenia susceptibility can be extremely helpful in identifying novel drug targets, disease biomarkers, and even help personalized medicine or help risk predictions. So that's the end of my talk, and I welcome any questions.